Hi, welcome back. My name is Preeti Balchandani. I'm a faculty member at the Translational and Molecular Imaging Institute, and I work on ultra-high field MRI. I'm going to be introducing the neuroimaging speakers, and our first speaker is Dr. Julie Price from the University of Pittsburgh. She's a professor of radiology and biostatistics. And her expertise is in quantitative PET methodology for translational imaging of protein targets, blood flow, and glucose metabolism, including studies of aging, neurodegeneration, neuropsychiatric disorders, and brain injuries. That's covering a lot of ground. She's the co-author of over 140 research articles. She's the head of PET methodology that includes PET-MR neuroimaging at the University of Pittsburgh, where she started in 1994. And from 2013 to 2015, she served as the chair of the NIH Clinical Neuroscience and Neurodegeneration Studies section. I met Dr. Price yesterday, and it was a pleasure talking to her. And I just want to thank her for some great advice on how to build a successful scientific career. And she's going to speak to us today about in vivo PET imaging of protein targets in Alzheimer's disease. So, Dr. Price, thank you.
Good morning. Can everyone hear me okay? Okay. First, I'd like to thank the organizers. I am honored to be invited to speak today. I'm going to talk about PET imaging of protein targets and try to provide a perspective of the development of the amyloid imaging field, and how rapidly it's developed based on the 30 years of applying PET to protein targets in general. Then I'll also briefly introduce the neuropathological hallmarks of Alzheimer's disease, which you're familiar with, but I'm still going to take us through it a little bit more just in the context, to provide a context for the studies that I'm going to review later. I'll go through some of the older studies that we did to validate the PIB PET, and then I'd like to just address how the field has progressed, because I think it's interesting to see 13 years later how data is being interpreted and how the field is moving forward. And then I will also just briefly talk about tau imaging studies done by colleagues at MGH in Berkeley. Okay, so moving right along here, one of the things I wanted to mention is it's hard to do PET imaging with protein targets. You have molecular specificity, that's great. PET is a very sensitive technique, but the radiotracers have to be of high affinity and selectivity for the target. They have to cross the blood-brain barrier. There are issues with polarity. When they enter the brain, you don't want metabolites in the blood or in the brain. You don't want metabolism of the radiotracer so that the radionuclide is no longer associated with your target protein, with your target radioligand. So there are many aspects that really go into thoughtful study design, and a lot of tracers just don't make it. And nonspecific binding is another issue, and some of this will come up in subsequent slides in terms of signal to noise. And the carbon-11 has a 20-minute half-life, and fluorine-18 has 110 minutes. And that means that if you're doing preclinical development, you can do multiple studies in a day with carbon-11. And with fluorine-18, if you're able to also label it with fluorine-18, then you can distribute it for more widespread use. And it's important, generally speaking, depending on the study design, but what I'll be talking about today is high specific activity so that you have tracer conditions. Okay, So this is just PET imaging examples that range from, I'm sorry, I'm having a little trouble here. That range from O15 water, which is a general functional measurement, all the way through to specific protein targets. For the serotonin and dopamine receptor systems. We recently wrote a review, Roger Gunn led this effort, and we looked at 6 categories of proteins studied with humans and PET over the last 30 years, and we classified them as ligand-gated ion channels, G-protein coupled receptors, which are serotonin and dopamine, and membrane transporters, such as the serotonin transporter. But also, studies have been done with enzyme inhibition studies or enzyme activity, and then the misfolded proteins, which is the focus of the talk today in terms of amyloid beta and tau, and also neuroinflammation with the translocator protein. So the next slide will just sort of put it in perspective in terms of how the misfolded protein or the amyloid field has grown and expanded rapidly. So initially, it was dopamine that was the primary protein target about 30 years ago. And in the next decade, PET continued to grow in terms of the G-protein coupled receptors, with some of the TSPO neuroinflammation work being developed and the dopamine synthesis. However, in the last recent decade, 2004 to 2013, there's almost 25% of the effort is now in amyloid, and so that field really has grown over the last 10 years. So just moving on, we'll just touch on some details about amyloid beta and neurofibrillary tangles. This is a slide from Steve Dukosky that I like because it just puts things in terms of a clinical perspective. Normal pre-symptomatic AD, mild cognitive impairment, and diagnosis of dementia. And the brain pathology just ranges from no disease symptoms, but asymptomatic, all the way through to substantial protein aggregation and accumulation in brain. And these things are all well known now, but this is something that really at one point was more established in neuropathology. And a theme throughout the entire last, I would say, 30 years or always for any disease is how early can you detect pathology? Can there be therapeutic interventions that actually make a difference if you go earlier? So just quickly in terms of the amyloid cascade hypothesis, what we're really looking at is accumulation of amyloid beta in terms of amino acid fragments of 40 or 42 amino acid fragments. And it's a sequential cleavage of gamma secretase and beta secretase and gamma secretase that produce this fragment. And the aggregation and oligomerization and fibril formation that leads eventually to neurotoxicity. And part of the amyloid cascade hypothesis would say that the formation of the neurofibrillary tangles, or the tau-containing protein, is a result of the imbalance of A-beta production and clearance. So support for this hypothesis is based on the rare mutations in chromosomes 21, 14, and 1 that code for the amyloid precursor protein. And the presenilins are important catalytic components of the gamma secretase complex, and that these mutations can lead to 100% penetrance for early onset autosomal dominant Alzheimer's disease. And then Down syndrome also arises from an extra copy of chromosome 21. And amyloid beta pathology is typically seen at autopsy. So all of these things form the basis. And then I just want to go through the distribution based on very painstaking work done by Brock and Brock over almost 25 years ago that really staged amyloid deposits in terms of initial basal portion deposition in the basal portions of the isocortex, and then it would progress through association areas, and advanced disease is fairly uniformly distributed. And the neurofibrillary tangles begin in the intraorbital cortex and spread limbically to the isocortex. So these are the things that people wanted to demonstrate with the PET imaging. And I just want to go through now, move forward in terms of the radiotracers that are more often used for amyloid imaging are 3 that are fluorine-18 labeled that have been approved by the FDA, technically to rule out evidence of Alzheimer's, but of course there are off-label uses as well. The NVIDIA compound here is a very good compound. You can see consistency in the distribution of for a subject scanned with all of these compounds. The NVIDIA compound is carbon-11 labeled as well as fluorine-18 labeled, and that will probably soon be FDA approved. In the meantime, many of the studies done worldwide were done with Pittsburgh compound B. So I'm going to take a few minutes just to take you through the development work that we did, not in great detail. It's going to be just a general presentation. So I worked with Chet Mathis and Bill Klunk, who actually co-invented the compound. But certainly one of the first steps was postmortem evaluation in AD frontal cortex. And I always like to show this because one of the first things to demonstrate is that indeed there's a high affinity component as you do the preclinical evaluation. And then the next step were to look at binding homogeneous assays, and the 6-hydroxy-BTA1, which eventually became PIB, was found to very highly label AD frontal cortex, but not the cortex of controls or non-AD dementia. And the cerebellum, which we now have adopted as a non-specific area, was seen to be fairly non-specific across controls AD and non-AD dementia. So all of this was very promising as we move forward, and then the ratio of frontal cortex to cerebellum for the AD subjects was quite high. So the specificity of the benzodiazole imaging agents were first described, and then we moved forward to state these basic facts. This was many years ago now, and we've learned more about this. But at the time, PIB bound to both A-beta plaques and tangles at micromolar concentrations, but binds only to A-beta at 1 nanomolar concentrations, and the PET studies are generally performed at nanomolar concentrations. And that, again, gives you an idea of the sensitivity of PET that we leverage. And it binds to fibrillar but not diffuse A-beta at 1 nanomolar and does not bind to alpha-synuclein. So it was fairly selective and specific. There were other ligands available at that time, but PIB was fairly specific. And then in collaboration with Uppsala University, a proof of concept study was done that shows the control uptake. It was pure uptake. It wasn't a quantitative study, no MR imaging, that demonstrated in a control the uptake of PIB was mostly in white matter. Again, there was still a nonspecific signal we knew we were going to have to deal with. And FDG distribution for the control was fairly consistent with control distribution of glucose metabolism. On the other hand, PIB localized very heavily in the AD subjects in the frontal cortex, and at the time, there hadn't been as much sampling of frontal cortex. Now we know, obviously one of the strengths of imaging is you can look through the whole volume. And I think that knowledge that has been gained from imaging has informed the neuropathology process and diagnosis as well. And the AD subject exhibited what at the time was a characteristic signature of FDG of bilateral hypometabolism in the parietal cortex. Okay, so moving on, then we extended, the next step was to extend the proof of concept studies and do quantitative imaging, do full compartmental modeling, sampling the blood, estimating quantitative variables rather than simple tissue ratios. We have to do this in order to verify the limits of the use of simple tissue ratios. It can be impacted by plasma clearance and nonspecific factors. So we then perform modeling for many subjects across controls, MCI and AD, and then we were able to progressively relate the quantitative measures to more simple measures like the Logan graphical analysis. And we were able to establish the level of bias as we progressed, because it's just not practical, obviously, to do full compartmental modeling. As we move forward, this study also was the first where we realized, and this is, again, absolute common knowledge now, but we realized that there may be, MCIs may have quite a range of amyloid load from control levels to levels that may be even as high as AD. So the next step then was to try to further simplify. And we're really simplifying to the level of tissue ratios and relating tissue ratios to fully quantitative measures. And when you're separating AD from control, it's pretty much anything can be used to separate them. It's the area, it's the intermediate area that still remains a challenge. And certainly, as we collected more data, we saw more overlap between AD subjects here in blue and the controls in red. And we began to see more overlap and a need for trying to establish methods to define PIB positivity and PIB negativity. I'm just describing work that we did, but these efforts were also ongoing internationally at this point. So one of the things that had been applied is a simple buffer zone area between PIB positive and negative based on people that we really believe were negative. But it just wasn't robust. I mean, the distributions, certain methods worked well when the negatives and the positives were well separated. But when you have a continuous distribution, particularly we saw in very old control cohort, 80 to 95 years old, that the distribution of SUVR values was much more uniformly distributed. And we then applied a k-means clustering method that we found really performed more consistently. And one of the things that we found was important was to also compare those results to consensus visual reads. And we found that there was agreement, and I think the reads helped inform the cluster process, and I think the clustering helped inform the visual read. I'm not going to talk very much about this, but this is probably something I've spent a lot of time on the last couple of years. But there's been a lot of work by many groups to look at postmortem correlations. And what I mean by that is relating the PIB, regional PIB, SUVR or DVR, which is a more quantitative measure, of accumulation to the amyloid beta load measured by autopsy evaluation. And we find the red boxes are studies done in our lab where certainly with an Alzheimer's disease subject, we found very high correlations, higher than we had thought at first, of almost 0.8. But then there are some subjects where the PIB was negative, but then there'd still be frequent amyloid deposition. And what we think is happening now, and I'm not going to really show this right at the moment, is that even some diffuse plaques can have fibrillar content. And if there's fibrillar content, the PIB binds to that. So there may also be some labeling of diffuse plaques. And I think that that's something that we're trying to understand in terms of what the differences between the amyloid pathologies in these various groups and the PIB signal. So quickly I'll go through progress in the field. Again, this just shows the range of PIB uptake. It was pretty clear that you can be cognitively normal and still have substantial accumulation of amyloid, what makes some people more vulnerable to maybe progress to mild cognitive impairment in AD. We're still working to try to figure that out, and I think one of the areas that people look to for that is the familial early onset AD. That's very different from sporadic AD that happens in the mid-60s and later. But I'm just going to show you some early results that we had when we looked at PIB in familial AD based on these genetic mutations. And this was also in collaboration with MGH. But it's very rare, but it's 100% penetrant. And this was Dr. Klunk's study. This is just for comparison purposes, a normal control. And a normal control after longitudinal. This was one of our first longitudinal subjects. But there is some evidence that there's an increase, but this is visually what we were dealing with while we were trying to quantify. But what we didn't expect was the very targeted striatal uptake in these subjects. And I think it was Dr. Klunk who thought that we had administered raclopride instead of the amyloid beta. But then we've also done studies, collaboration in Down syndrome, and they also show high uptake in striatum, consistent with the thought of maybe overproduction in the mutation subject cohorts as opposed to impaired clearance that may be more consistent with sporadic AD. So several groups have looked at longitudinal follow-up, and this is an area where I think the particular binding measure can make a difference. I do think that that we're still working to try to make sure that that some of the noise and some of the limits in terms of the non-specific component is not interfering. We're going to have to, it's going to take time to really understand, I think, some of the things that we're seeing with these longitudinal studies, such as this is work by the Australia group where there's an increase in change in SUVR or amyloid deposition per year, and it peaks the peak change happens at an amyloid burden of about 2.3, but in some it plateaus. For some groups it plateaus, but it generally decreases. And we've been trying to, this happens in most labs, and we're trying to understand if it's, there could be several factors for that, but it seems that there may be just a ceiling to amyloid accumulation and that's beyond what would, partial volume correction or atrophy is not would not explain what people are seeing across groups. And then it clearly declines with the MMSE as well. And this is images from an FAD, a familial AD subject scanned many times, over 8 times over, I think about 10 years, 10 or 11 years. And we don't see remarkable accumulation at later follow-ups. So it's consistent with that leveling off, even in the familial AD. So I'll just quickly go through this, but the Australia group has imaged hundreds of subjects, 400 over 4 to 5 years. And one of the things that they at first hypothesized, or they looked in the literature, and the post-mortem prevalence of plaques in healthy controls was right in line to the prevalence seen with the PIB, which are the blue circles plotted over the green triangles, which is the post-mortem. And then the prevalence in AD would suggest a 15-year development period. Since that time, they proposed this as the model of the natural history of A-beta deposition in sporadic AD, where the MCI and AD have higher levels of amyloid compared to the healthy controls that are positive. But their data suggests that that it takes about 12 years to go from a negative level for the SUVR to a level of 1.5, which is their cutoff between positivity and negativity. Then it takes another 19 years to go from threshold of positivity to full-blown accumulation associated with dementia. So this sigmoid type of shape is what is driving their data, but I think more studies in the autosomal cohorts will further inform this. And that brings me to, I just want to bring up a couple of multi-site collaborations that I think have really impacted the field. One is the ADNI. Clearly, we all are aware of it. It's really impacted the field. And in terms of biomarkers, I think one area in AD is the work by Cliff Jack at Mayo Clinic, where and Risa Sperling, several researchers contributed to this, but Cliff was one of the first to publish it in terms of the early amyloid accumulation, amyloid coming first, synaptic dysfunction, tau-mediated injury happening next as you progress from preclinical to dementia, and then the brain structure, the hippocampal atrophy coming later, and cognition coming at the end. So clearly, it's earlier treatment is needed with all of these brain changes that are happening. And now people are thinking more in terms of amyloid positive, amyloid negative, neurodegeneration positive, neurodegeneration negative. And the neurodegeneration are the FDG PET measures, the tau CSF measures, and also functional connectivity measures, or fMRI measures, are the outcomes that are associated with the neurodegeneration as opposed to amyloid. And I say that because I think that what's happening now, and this is a 2016 publication by the Mayo Clinic group, where they're now estimating rates of biomarker states by splitting subjects into these categories where you may be amyloid positive, neurodegeneration negative. And they've had to do it because one reason they were motivated to do it is because of the amount of variability in the data as they were trying to work through different models. So I have a few more slides. These are other multi-site studies that are ongoing, and these involve therapeutics that I think will also inform the field. And this is the large early onset AD study and the A4 study that's being spearheaded by Risa Sperling, and the Banner-initiated study to examine extended family members in Colombia, as many share a rare genetic mutation that really triggers Alzheimer's symptoms. So these studies, I think, over time are really going to further inform. And just quickly, we're also working on standardizing these measurements across scanners and sites. And it's a multi-institutional effort and industry effort as well. So what about tau? I'm going to wrap up real quick. You know that tau is important for neuronal microtubule integrity, and hyperphosphorylation leads to reduced tau binding, loss of tau, sequestering, aggregation. And I show this because I think this is a great review article. We've talked about requirements for tau tracers, but for tau particular, you really need high selectivity over A beta. And that is a big challenge for tau imaging, plus the fact it starts out intracellular, but I think it then, upon cell death, can be extracellular as well. There are many isoforms, 6 isoforms at least, with different conformational structures, and that also can we're trying to understand tau imaging that results in tauopathies that may that are complex compared to the mixed isoforms that we see in AD. So this is another area where I think maybe even more ligands with more specific conformational selectivities will be useful in the future. And there's several ligands. T807 or AV1451 is the most widely used. The MGH group has demonstrated that even with that there's clearly off-target binding in areas such as, we call it off-target binding, in areas such as the putamen, but nonetheless, the binding is consistent with MMSC decline, there's a tighter relationship between cognition and tau binding than certainly was seen with amyloid. And the Brock staging has been successful. The Berkeley group has shown they actually generated regions of interest that were specific for each of the Brock stages, and they've really been able to demonstrate on the first pass that even with the larger distribution of one of the other higher risk factors for AD, that the Brock stage is increased with ApoE frequency, and mini-mental score is also consistent with the higher Brock staging. And again, hippocampal volume decline is also consistent with what they've seen with their surface projections of the AV1451, despite off-target binding that was mentioned in this article in terms of choroid plexus. So this is a neuropathology article that addresses the neuropathology results for T807, and this is important also in terms of the striatal uptake that is not associated with tau. So I'd just like to thank my collaborators, certainly Chet and Bill, who invented the PIB and provided me with a great opportunity to work in this area. And also, I wanted to thank the volunteers and their families, and I also thank you.
Thank you so much, Julie. Does anyone have any questions? You can come up to the microphones on the side.
So very interesting talk demonstrating the potential of PET imaging for neuro applications.
Even though we have blurry images and a lot of motion.
Yes, but that's part of my question, actually. I wanted to ask you, you have showed some PET-MR fused images. So the first scale of my question is, have you evaluated the benefit of using MR with PET for the dynamic PET imaging that you've studied? And the second question, whether you have thought of combining different tracers as a dual tracer study to maximize the benefit of PET imaging for these applications.
Okay, I'm not sure I understood exactly what you were asking in the first question, but I definitely understood what you were asking in the second. Have we incorporated MR information into our PET analyses? Is that what you're
PET
asking?
reconstruction and analysis to correct for motion
like
and partial
priori.
volume.
We have not done that. I wish we would do more of that, but we haven't. And we obviously could benefit from doing, from trying to do some of these things. And with the PET-MR, we've been talking about how to do this, but we really have not. That's not a strength at the moment, but that is something we would want to do. Yes, I've certainly thought about dual tracer studies. I think that the early studies, I think Bob Kepi at Michigan was most successful in trying to do that. I think it was flumazenil and I can't remember the other ligand. But it would be very useful provided you have radio tracers with appropriate kinetics. The T807 is just, it has very complicated kinetics, particularly, in my opinion, for high binders. I think that there's a real challenge in really quantifying. The thing is, you know, the tissue ratio is a very important and feasible measurement for disease populations, but it has its limitations. And I think for T807 and high binders, that's true. But some of the more rapidly equilibrating compounds, some of the maybe THK compounds, may be amenable to dual tracer. But then carbon-11 is probably desirable, and that might not be as easy for everyone.
Okay, thank you.
So maybe I have a question. I guess people can hear me. So you mentioned when you're describing the disease itself, you have basically said there will be deposit of beta amyloid and also tau, and then really it's neuroinflammation and then cell death. Would that be important then to image neuroinflammation? Is it why would that be less of important than why are we after the beta and the tau at this stage?
Okay, what I would say to that is we're after the tau and the amyloid beta because we know that those are abnormal protein accumulations and neurons die. We also know neuroinflammation is very important, but we still are in need of really good TSPO PET ligands. Now, PBR28, I think Bob Innes' group and Dr. Kreisel, who's now at Columbia, they've done studies that have started to distinguish neuroinflammation role, but it's complicated in terms of where in the disease trajectory you study people. One last thing. The other thing that makes it complicated is the polymorphisms and the sample sizes that are needed in order to do some of this.
Actually, Paul Matthews gave a talk here about maybe a year ago or less, and he talked to us a lot about this. So what you're saying is because we lack of a good tracer, and if we have a tracer that can do neuroinflammation, then we should be better than maybe the A beta and the tau?
Well,
Maybe.
I don't, maybe. It also, it's obviously complicated. The interplay between all those factors.
So when you're using cerebellum as a baseline, what we can use for tau imaging, because sometimes in tau imaging, even cerebellum is getting hot.
Well, sometimes in amyloid imaging, cerebellum can have diffuse plaques, and if diffuse plaques have a fibrillar component, we're still, kind of locked in a little bit. So I think what we try to do is we depend on neuropathology to guide us as we do more and more of these studies. Maybe we'll understand better what areas. But I would say to that is that we're using cerebellum, I think, because we believe that in the subjects that we're studying initially, that it shouldn't be as impacted by the neuropathology. Do you agree with that? In these very early, or in the,
Yeah.
I mean, if we go to some of the more, like maybe PSP, although that's a complicated situation there, but maybe with some of the more tauopathy, specific tauopathies, it might be more complicated provided T807 binds to that.
So when we do the tau imaging, that's still questionable. People are discussing how to quantify the tau.
I do think so. Yeah. And I think it also depends on the ligands are sort of similar. Right. But there are different properties to these ligands, and the ones that slowly equilibrate, it's even harder. And so I think these are all conversations that we'll have for a long time.
Thank you.
Hopefully not too long, but.
There is an argument or dispute about the spreading of tau pathology or even amyloid. I mean, from specific area to spread,
Did I do that okay?
My question is,
Yes.
This PET imaging
Yes.
can help to understand or resolve this issue. I mean, it's really spreading or...
Well, I think again, I sort of mentioned it before, I think, and we all know this, but I think what neuroimaging really helps to do is for you to see the whole volume. And as you image across the spectrum, do repeated imaging, longitudinal imaging, I think it does help inform the general process of this spreading and where it originates. I think we still follow Brock because clearly that was a carefully done study. But as we move further and further toward asymptomatic, very, very early, and people really want to push it very early, I think that that's where the imaging will help us learn more about how it propagates or accumulates.
So to thank you for your beautiful talk. This is a present.
Thank you.
And do you want to?
Thank you.